All right, we are recording. So today is April 19th. So you did have a quiz due this morning, chapter 10. Um, and then only one left, chapter 11 quiz, which will be due a week from today. So I'll post that this afternoon. This will be the last quiz I've mentioned before. I'm going to be canceling chapter 12 quiz, so that will never happen. We do still have the Master's Emissary, the main writing assignment is due currently Wednesday. So the 5th of May. So this Wednesday will be the 21st. 2 weeks from this Wednesday will be when the master and his emissary paper is due. I have posted several of those lectures that cover several parts of the book, like the first half of the reading so far. I'll definitely post some more of those lectures here this week. But yeah, the paper's due 2 weeks from this coming Wednesday. So I'm still happy to talk to people about their project or their topic ideas. If they would like to kind of flesh those out with me, feel free to send me an email and we can do that. Any questions or comments about the schedule, things coming up here and the next, the final few weeks of this course? Any questions or comments? All right, apparently not. So we will finish talking about attention. We'll see if we can start talking about chapter 8, which is about the motor system. So let's finish talking about attention. So yeah, I do want to talk about, you had those in-class questions last time. Let's find and talk about one of those real quick. Well, maybe we can talk about all of them real quick, actually. So what is the difference between frequency analysis and event-related potentials? Frequency analysis also involves a Fourier analysis, just breaking down the electrical activities measured from electrodes on the scalp into those different sinusoidal or wave components, such as delta, theta, alpha, beta, gamma. That's frequency or Fourier analysis. And then event-related potentials, you're still using those electrical signals recorded from electrodes on the scalp, but you're not performing any Fourier analysis. You're not breaking down that electrical signal into the different sinusoidal components. You're just measuring kind of the raw electrical activity, usually at a given window of time after some event, and you're measuring that electrical activity without breaking it down into its sinusoidal components. It's just the raw kind of electrical signal at some specific window of time after some event occurs. And we dealt with an example, by the way. Where is it real quick? Yeah, the P1 is the one that we've talked about a good deal. So it's a good idea, for instance, to know, maybe we should ask for this reflexive cueing study, what happened at time point 0? Think about this real quick. What happened at time point 0? What was the thing that happened at time point 0 in this study? You want to remember what was presented or what happened at time point 0 in this study? This P1 event-related potential, this is the brain's response depending on whether a person is paying attention to something or not paying attention to something. That something is right here, but what is presented at time point 0 in this study? What is the thing that occurs? This is an event-related potential study, meaning we're measuring the electrical activity at some specific window of time, P1. Here, that specific window of time is the first positive peak. It's around 100, maybe 200 milliseconds after something occurs at time point 0. I'm not seeing any guesses. Oh, here we go. Maybe I am now. The white rectangle. Let's take a look. Yes, the white rectangle, which that is the correct answer. What is the white rectangle in this study? And I'll give you a clue. The word is written somewhere on this slide. The white rectangle is the what in this study? Let's see. Yes, the target. We had several people who got the target. So the white rectangle is the target. And at the time point 0, this is the presentation of the target. That white rectangle, which is the target, that is the thing that occurs at time point 0. And so the researchers, this is an ERP study, they're measuring the electrical activity that occurs on the scalp in this window of time, this kind of surrounds the P1, the x-axis here. This is time in milliseconds. The P1 here, it looks like it's around 100, maybe 200 milliseconds after the onset of that target stimulus. Each one of these lines is probably 100 milliseconds right there. Yeah, it is. Oh, each one of these lines is 50 milliseconds actually. So 50, yeah, 100 to 150. The window right here is between 100 and 150 milliseconds after the onset of that target. You can see that the P1 response is bigger to the dotted blue, which is the attended target, when you are paying attention to the same side of space that the target occurs. For the short ISI, duration between these two is only 50 milliseconds. You get an enhanced P1 for the cued or the attended target. For the long ISI, that is when the duration between these two is 250 milliseconds. The zero time point, it's still your brain's response to that target right there. But the amount of time between these two things is long ISI. You get kind of the opposite pattern where the ERP response is actually bigger in response to the not attended target, the uncued, the red. You can see the red goes further down here, but it's the blue that goes further down here. And this illustrates that concept talked about in your textbook, and I talked about here, called inhibition of return. Real quick before I move on, the name, what is the name of this thing right here? These little 4 dot things. What is the name of that thing right there? Yeah, Q. That is correct. It's Q. And this is a reflexive cueing situation because this Q, these little 4 dot things, or these 4 square things, are meant to kind of steal your attention. They surround they blink real quick and peripheral vision, which kind of steal your attention over, presumably, to that side of space. And you get this attentional enhancement when the amount of time between the Q and the target is short. But you got to get this attention inhibition, or attention kind of hurts your attention in some ways if there's a long ISI here. As the case here, you actually have bigger responses for the target when it's not in the attended visual field for the long ISI. That illustrates this idea of inhibition of return. Anyway, I want to go back to these questions. It's a little longer on that than I planned. There. Okay, so what is the difference between endogenous and exogenous attention? Endogenous, inside, this means voluntary attention. The Q in voluntary, so I'll say it again, endo means voluntary attention coming from within your own mind. So can someone go ahead and write in the chat box, what is typically the attentional cue in an endogenous attention study? I think every example we saw of endogenous visual attention, there is the same type of cue, visual cue was used in all endogenous, that is voluntary attention studies. What is the attentional cue in these endogenous, that is voluntary attention studies? Let's see how we're doing here. Yep, it is that arrow. Compared to exogenous attention, it was those 4 little dots, or those 4 little squares that kind of blink on the screen. And the difference, once again, endogenous, it's like you're in control of it, it's voluntary, versus exogenous, it's more like a reflex. The thing which is something out there is pulling your attention to one direction. So endo, it's coming from within, the attentional, the decision to attend to different things. And exogenous, you don't really have control over the decision. Something's kind of pulling your attention. And then I would say the most important question right here, the final one, how are the concept of extinction and biased competition similar? This is a tough question, but it's getting at kind of a big picture theme in this chapter, which has to kind of the main similarity between concept of extinction and biased competition is the term is actually the concept of competition. Right there, competition is the main similarity, and I will explain by going back to this stuff over here. Competition. So here's an extinction. It's that behavioral example. It kind of helps explain those situations in an individual with visual neglect. Those situations in which they're going to have the most trouble paying attention to something in the the visual field contralateral to the brain lesion that they have. So usually the brain lesions for visual neglect are in the right hemisphere, which cause visual neglect of the left visual field. However, right here, this individual, this is the individual with neglect, is able to see something in his left visual field. And the reason he can see something here, but he's able to attend to his left visual field, is because in this situation A, there's minimal amount of competition. There isn't a lot of noise. The scene in which he is, visual scene in which he is embedded is not cluttered, not complicated. There's not a lot of competition. But here in this last scene, he's no longer is able to notice what's going on in the left visual field. He's no longer able to attend to his left visual field here. And that's because there is competition in the other visual field. It's kind of like whatever happens in this right visual field kind of steals his attention and makes it so that he can't devote his attention to the left visual field. So in extinction, and it even says it right here, competition is like a really fundamental, is the fundamental thing which causes you to be able to see the attentional problem in these individuals. You don't really notice the attentional issue until you embed them in a scene that's complicated where there's competition. That's the basic definition of extinction. You don't see the impairment unless there's competition among at least these two stimuli. The one in the right visual field right here, the one in the right visual field interferes so that the person cannot attend to the left visual field. That only occurs when there's competition. And that's a similar type of thing in this biased competition model. This is where we left off last time. In the biased competition model, it's kind of described nicely down here. The attentional effects in the brain are biggest when the scene in which you're making the person pay attention to is kind of cluttered, noisy, when there's a lot of competition in the scene. So the attentional effects are biggest when there's competition. That's the key similarity between extinction and biased competition model. In both situations, the attentional effects or the attentional impairment in the case of neglect only become obvious when there's competition, when there's a noisy scene. And so here is the study we talked about regarding biased competition model. There's these two conditions, sequential, in which you're seeing each of these things, each of these little colorful square things by itself. In this condition, there is no competition because you're looking for one of these things. One of these is a target, but each time one of these things appears, it appears by itself. So when each one appears, there is no competition from the other colorful square thingies. However, in the simultaneous condition, all four of the colorful square things appear simultaneously. This is the situation where there is competition. You are looking for one of these things that's the target. When that target occurs, it's simultaneously occurring with other ones as well that are not the target. Those other ones are the things providing the noise, providing the clutter, providing the competition. So you only get competition in this simultaneous condition of this experiment. And so this is where we left off. These figures, which are a little bit tough. I started explaining them last time, and I will kind of explain them again here. So what's the height of these little traces represent are activations in two different parts of the brain. V1, primary visual cortex, and V4, another region kind of in the occipital lobe, but it's further along in the visual pathway. V1 is activated first. V4 is activated a little bit later. And they're interested in how competition influenced these brain responses of V1 and V4 in this study. An important thing to remember about this figure, which if you read the textbook or I think the notes say it as well in the slides, these blue stripes right here, these are the conditions in which the participants were asked to pay attention to one target in the scene. They would see these colorful squares appear on the screen. And in those conditions where there was blue vertical lines here, those are the conditions where they're asked to do that, where they're asked to focus on one colorful box thing during each trial, compared to over here when there is no blue vertical lines. Here you can think of the participants just kind of passively viewing the colorful squares on the screen, but they're not trying to find one. They're not looking for a target. Okay, and so a key difference, there's a couple things that we talked about last time. And the most important, as far as the biased competition model, is the fact that you only get these attentional enhancements when there's competition. When there's not competition, when there's not noise in the scene, the visual system and the brain are not taxed enough that paying attention boost the way the brain works in any notable way. But when there is competition, that's when you notice the enhancements from the brain. They actually help with the brain signal. And you can see that by comparing the blue traces right here, the ones with blue vertical line, to the ones over here. This is when people were paying attention. This is when they're not. And these these conditions, sequential, simultaneous, sequential. And if you notice in V1, all of these conditions, the brain activation is basically the same. And this is a pretty important concept according to your textbook, and actually in general in cognitive neuroscience of perception, is that V1, there's not a whole lot of attentional modulation in V1, no matter what. And you can tell that because when people are paying attention, the black traces are basically the same height in these same two conditions compared to when people are not paying attention to them. So attention doesn't do anything in V1, but attention definitely does something in V4, particularly for the simultaneous condition. So here's V4, and the simultaneous condition, that's this blue thing. Here's the simultaneous condition when people are not paying attention. Here's the simultaneous condition where people are paying attention. And you can see that there's a difference right here. The brain activation in V4 is bigger in the simultaneous condition when people are paying attention compared to when they're not. However, for the sequential, and this is the thing I didn't show you last time, so this is a new but important nuance regarding these findings. In V4, for the sequential condition, you have to compare kind of these two things right here. Sequential condition when you're not paying attention. So this is when each colorful square is by itself and you're not paying attention to anything. This is the brain activation in V4. This is the brain activation in V4 when you are paying attention to the sequential condition, looking for a specific target. You notice there's not really a difference in the height of these two. And that's because in the sequential condition, there's no competition. There's not really noise. There's not really clutter when the target is being presented. So you don't get any attentional enhancement, but you do get it for the simultaneous condition of V4. There's a difference here. You see it in the simultaneous condition. That's one where there's clutter, where there's noise. You see the attentional enhancement in V4 for the simultaneous condition, but you don't see attentional enhancement for any other conditions. This is really the key finding right here, is that in the simultaneous condition, when you attend to it, it boosts the activity in V4. You don't find any boost in V4 for the sequential condition. So the two findings, once again, for the studies, it's kind of a tough one, I think. The attentional effects are biggest when there's competition. That's the simultaneous condition. And you really only get attentional effects in V4, at least in this study. You don't really find that many attentional effects in V1. And this is kind of seems to be a trend in cognitive neuroscience of perception, is that attention seems to have more of an influence on later brain regions than it does earlier brain regions. So attention doesn't do much in V1, but it does more in V4 because V4 is a relatively later brain region. So here's some more evidence kind of about this idea that attentional modulation tends not to be that big in V1. Those very early, more like sensory areas of the visual system do not get a whole lot of attentional modulation. So this is an fMRI study. And these are regions of the brain that were modulated by attention. So these, in this study, I think people were paying attention to something either in the left visual field or they were paying attention to something in the right visual field. And they wanted to see, how does paying attention to one visual field or the other influence activation in the contralateral hemisphere of the brain using fMRI. And so they found kind of two little chunks of activation that are bigger when you're paying attention to the contralateral visual field compared to when you're not paying attention to it. And the key ones that I want to talk about right now are these that are traced in black right here. And what's important to note is that these are visual cortex. You can see that this is right here. This is the occipital lobe. So this is back here in the brain. So this is visual cortex, but primary visual cortex, which is important, Primary visual, this is like a map of visual cortex right here. V1, primary visual cortex is about right here, or in this, on these brains, primary visual cortex V1 would be about right here. It would be about right here as well. So an interesting thing to notice is that there is attentional modulation here in these visual areas, but V1, primary visual cortex, appears to not be one of them. The green area, V1, primary visual cortex, is right there. And there's really not any attentional modulation of that particular brain region, V1. So the brain activation, or the enhancement by attention, is these other relatively later brain regions. I mean, I've mentioned this before. So V1 is up here, V1, and then the visual information, the visual information, it reaches here first, and then it kind of goes down, and then along the ventral pathway, at least in this direction, it's going from the occipital lobe to the side, to the temporal lobe. And then over here, same thing. We have the occipital right here, and the information starts in the V1, goes down to these later visual regions like V2, V4 down here, before going along to the temporal lobe, the ventral pathway over on that side. So it goes to each side of the brain. But the most attentional modulation occurs in these later visual regions like V2, VP, V4. These are later than V1. And these are the regions which are highlighted here, that are traced here in black. These are later visual regions, not V1. These are also later visual regions, not V1. So this supports that same idea from the previous study. And this is something kind of repeated in several different parts of the textbook. Attentional modulation tends to be bigger for later you get in visual processing, the more likely you are able to see or measure attentional modulation in that region of the brain. The early sensory processing regions, not quite as much attentional modulation compared to the later regions. And that's shown in this study too. And I'm going to skip over that for time. So here's another region of the brain that was modulated by attention in that same study. So once again, people were attending to the left or right visual field. And they also showed these activations right here that were bigger when people were paying attention versus not paying attention. These activations, though, are part of the lateral geniculate nucleus, which is part of the thalamus. And we've talked about this. But I'll show you here on this little brain. The eyes are up here. And if there's something over here in the left visual field, that information will cross into each eye and then find its way primarily to the right hemisphere. And then visual information, kind of the first area it goes in the brain, is right here, which is where the lateral geniculate nucleus of the thalamus is. And some of that most of that information after the thalamus will go to visual cortex right here. But before, and I have mentioned this as early as chapter 4, the first kind of place in the brain the information goes is thalamus right here. And there's also a stop in the brainstem right here. This all happens before that information reaches visual cortex. But you do see attentional modulation in the thalamus. And so I kind of think of the thalamus in this role as kind of like a gatekeeper. Depending on whether you're paying attention or not, the thalamus can be thought of as like a gatekeeper that allows information to reach visual cortex when you're paying attention to it. But it allows less information to get to visual cortex when you are not paying attention to it. LGN is modulated by attention. You can see it right here. When you're paying attention to a stimulus, there's more brain response in the thalamus compared to when you're not paying attention to it. And so basically the attention that you provide to something allows information to get to visual cortex. If you're not paying attention to it, relatively less visual information gets to visual cortex. So it's like, I think of it as like an open or closed gate from the thalamus that kind of prevents or helps information get to the visual cortex right here, which is where V1, V4, all of those different brain regions are. And I do want to say this because this is it's kind of a complicated thing, but it's important because we haven't talked too much about inhibitory processes in the brain, but this is a good example of one right here. So these are two different types of neurons that exist in the thalamus. Once again, the thalamus, this region right here, visual information travels into the brain. One of the first places it goes is the thalamus, and then also the subcortical regions right here at the same time before any of that information gets to visual cortex. And there's these two different types of neurons in the thalamus. Let me first look at this question or comment that we have here. Yeah, well, so the question is, without this thalamus function, would the visual cortex get overwhelmed? Yeah, I think that makes a lot of sense. I mean, it's very rare that a person would have like their thalamus completely not functioning. I couldn't even really imagine what would happen if that occurred. But yeah, I would imagine you can think of this thalamus function kind of like the gatekeeper. Is that, yeah, it prevents the visual cortex from being so overwhelmed that it wouldn't be able to make sense out of anything. Only that information which is attended kind of gets through to visual cortex. And I like that, the idea that prevents visual cortex from being overwhelmed. And so there are these two different neurons in the thalamus. The LGN neurons, these are excitatory, and I'll show you why I mean that here in a second. But then there's these other types of neurons also in the thalamus, and they're inhibitory. These are thalamic reticular neurons, TRN neurons. These are more inhibitory. And I said these LGN neurons are excitatory. And so what I meant by excitatory is that these LGN neurons, they can allow information. If you're paying attention to something, that boosts the activity of these LGN neurons, which causes more excitatory signal to reach visual cortex. And you can see that that's what's going on here in the LGN. The dependent variable is the amount of action potentials in specific neurons in this part of the LGN, this part of the thalamus. And the dark trace is bigger than the dotted trace. The attended condition, attend in, when you're paying attention to a stimulus, the response of that stimulus in the thalamus generates more action potentials compared to when you're not paying attention to it. So this is kind of the classic enhancement response that we see when you're paying attention to something. When you're paying attention to something, these LGN neurons, their activity increases, which allows more excitatory signal to reach visual cortex. However, there's these other kind of inhibitory neurons, TRN neurons, thalamic reticular neuron, which do just the opposite when you're paying attention versus not. I kind of think of these neurons as like they're kind of constantly providing brakes to the input to visual cortex. And you can see the brakes right here, because when you're not paying attention to something, these thalamic reticular neurons, TRN neurons, when you're attend out, not paying attention to something, there's more activity of these neurons, more spikes per second, more action potentials occur in these thalamic reticular neurons when you're not paying attention to a stimulus. When you start paying attention to a stimulus, attend in, the solid trace, the activity of these neurons decreases. Kind of the net effect of all of this activity in the thalamus is that when you're paying attention to something, more information goes to the visual cortex compared to when you're not. And that involves both of these types of neurons, these excitatory neurons, which cause more excitation to be sent to V1 when you're paying attention to something, and these inhibitory neurons, which their activity becomes reduced when you're paying attention to something, which kind of lifts the brakes off that information getting to V1. There's constantly always a certain amount of brakes being applied so the information isn't overloading V1. When you're paying attention to something, the brakes in TRN are actually released, which allows more of that information to get to V1. So this is kind of some nuanced activity of different neuronal populations in the same brain region. And this is just kind of giving you a little glimpse into how cognitive mechanisms in the brain appears to be quite complicated, involving interaction of both excitatory and inhibitory components. A couple more things on attention. We're almost done here. So this is a concept, which is, I remember talking about it a lot in graduate school, and it's like a bigger picture theme of kind of attention research in general, which is that there's very many different types of ways that you can pay attention to things that are out in the world. In this chapter, we've been focusing mostly on the visual system. We've talked about the fact that mostly what we've talked about visually so far is the fact that you can pay attention to different parts of space. You can either attend to the left side of space or the right side of space. So that would be that type that we've mostly talked about so far for visual attention would be visual or spatial attention. The cue directs you to points in space. However, in the literature on attention, there's actually many different types of ways that you can pay attention to things. And the type which is talked about here is feature attention. And so the two features here that we're going to talk about for this chapter have to do with V4 and V5, which are these two visual regions. V4 is known to process color. V5 is known to process motion. We've talked about these a lot. These are both in the part of the occipital lobe and visual cortex, but they're both relatively later visual regions compared to V1, and each is connected to different features. V4 has been connected to the ability to perceive color. V5 is the ability to perceive motion. And they do studies where they have people pay attention to either color or motion. And that's what this one is right here being described in this cartoon right here. Participants are asked to pay attention to either the color of a target stimulus or the movement of a target stimulus, whether it moves or not. And then different conditions, you're paying attention to the different either motion attention or color attention. They look at what parts of the brain become more active depending on the type of attention that you are giving. And not really that surprising given how I've set this up. When you're paying attention to motion, they see more activation in V5 compared to when you're paying attention to color, in which case they see more activation in V4. And so this illustrates feature attention, those parts of the brain that represent a given feature. When you are using your attention, one of the theories is the way that the brain works is by increasing the activity of those brain regions that typically process that feature. When you're paying attention to color, activity in V4 increases because V4 is the region of the brain which is known to process color. When you're paying attention to motion, activity increases in V5 because V5 is the region of the brain which is most involved with processing motion. So that's feature attention. And I guess we have, a couple more types of attention here. Vigilance. So this is something else which is talked about in the Master and His Emissary. It's talked about a little bit in your textbook, not a ton. Vigilance, though, this is definitely talked about a lot in Master and His Emissary, and it's an aspect of attention, though it's kind of an interesting aspect of attention in the sense that you're paying attention to kind of thing, McGilchrist called it open attention. You're waiting or paying attention to the possibility of a range of things that could happen. Oftentimes with vigilance, the type of attention would be deployed for kind of looking out for predators is one of the main ways that vigilance attention is thought to be deployed. You're kind of constantly waiting if you're an animal that has a tendency to be prey. When you're going out there to get for food, for instance, part of your brain is kind of scanning your peripheral vision, scanning the horizon for potential threats or potential predators using vigilance, kind of this maintained attention for something that could occur somewhere out there. I don't know what it's going to be exactly. I don't know where exactly it's going to come from. Vigilance is kind of a baseline state of readiness for things that can appear kind of unpredictably in space, and what the thing occurs, what kind of predator occurs can be unpredictable as well. So that's kind of vigilance. Another way of thinking about vigilance is anytime you're using brain power to keep processing or doing like the same task for a long time. So if you're having to deploy attention to a task for a long time continuously, that's vigilance. Oftentimes, as I mentioned, it's used for the task of waiting for or trying to find potential predators before they find you. You're kind of on this task of stimulation where you're looking for something in your peripheral vision for a long period of time. Vigilance. And there's some brain things about the vigilance. Some classic brain findings right here, actually. A good example of vigilance, classic example, is radar operators, like in World War II, where these radar operators have to look at a radar, potentially finding enemy planes or enemy ships, enemy ship boats. And obviously your job is to find, as the radar operators, to find those enemy planes that come into the radar as soon as possible. So you're kind of constantly waiting for this potential threat. You're looking at the radar, you're scanning it. And your job is that as soon as you see a threat appear on your radar, to announce it to, your superiors or ever, so that, or whomever, so that you can act according to that information as quick as possible to keep your chances of survival, to increase them. And they find that this type of task, usually performance is good in vigilance tasks for about 15 to 30 minutes. And this amount of time is apparently used even, for a job today, which requires vigilance, which is lifeguard, where if you've noticed the lifeguard, they're kind of waiting for a potential bad thing to happen as they're sitting up on their chairs. And they don't know where it's going to occur. They don't know the exact thing bad that could potentially happen, but they're kind of waiting in vigilance for that thing to potentially happen. And then the 15 to 30 minutes, though, comes from the fact that usually the shifts for lifeguards are short. I believe they're 30 minutes. If there's a lifeguard here, feel free to tell us about how long your shifts tend to be. But the idea that for most lifeguards that I've heard is the shifts tend to be short, 30 minutes max, because after that, vigilant attention, the ability to kind of deploy your attention kind of in a continuous fashion to find potential things that could happen, that ability is reduced after about 30 minutes. And so that's why there's quick changes in the shifts for lifeguards. Looks like we do have a comment here. Yep, around 30 minutes. Lifeguards here rotate it every 20 minutes and take a break after every 2 rotations. Yeah. So we'd have 2 different lifeguards apparently that kind of agree that every 20 or 30 minutes or so you change, you take a break, or someone else comes in, and that's to prevent you from becoming, your brain becoming so mushy that you miss something that's happening out there in the water. This keeps you sharp. You're sharp for about 15 to 30 minutes. After that, you kind of lose it. And there's a neural correlate of this related to frequency analysis. And you're best at vigilance tasks when high frequency waves are prominent. So here, beta, which is considered the second highest wave frequency, but there's also gamma. Beta and gamma are the highest frequency waves, so really fast moving waves. When those are prominent in your EEG, when you have a lot of high frequency activity, that tends to correlate with good performance in vigilance task, meaning you notice when there is a bad guy that appears, and you notice the bad guy fast. Theta is a low frequency. It's close to delta. Delta and theta are the 2 lowest frequency waves. When theta or delta amplitude becomes prominent in your EEG, that is when low frequency activity, it's kind of like, you can think of it as you're tired. When the low frequency waves get prominent, like theta, your ability to see targets, the quickness to respond to targets is not quite as good. So in general, when you're performing a vigilance task, I think this makes sense. You want the high frequency activity. This is what vigilance looks like. It's a lot of high frequency activity. When the activity in your EEG gets a little sluggish, that's when you're getting a little sluggish as well. So vigilance activity in general performs better at detecting targets when you have a lot of high frequency activity, which to me makes intuitive sense. And here's just some examples of some of the abstracts. Actually, I think these were taken from Master and His Emissary, which kind of highlight some of the findings regarding vigilance, which are talked about, which I always thought were very interesting findings from Master and His Emissary. These dual task studies in chickens and different types of birds, but I believe mostly yet lateralized chicks. They have the chicks performing 2 tasks simultaneously. One, they're looking for food grains amongst pebbles. So they want to eat the food grains, the rocks, the pebbles they don't want to eat, but they look similar. So one task is find the food grains and eat them without eating the rocks, which are kind of in the same area. That's one task. The other task is look out for a predator, like a snake or something like that. And so these chickens, they have them to do these 2 tasks, presumably like the researchers somehow provide a potential threat of a predator. So they must have a snake cage or like a picture of a snake somewhere, and they could potentially produce this around the animal to make the animal think that there's a predator in the area. So the animal, in this case, the chicken, performing these 2 tasks: finding food, avoiding getting eaten by a predator. And then they look and they find consistent uses of different eyes in these chicks, depending on the task that they're performing. In general, these chickens use the right eye to look for the food pebbles, the food grains rather. So the right eye supposedly connects in these animals more to the left hemisphere, and the left eye presumably connects more to the right hemisphere. So this kind of is an interesting, the support for the McGilchrist findings. The left hemisphere appears to be good for that kind of focused attention, looking for the details, uh, the food grains, finding them amongst the food pebbles involves mostly that kind of analytic attention, detail-oriented left hemisphere type of attention, whereas vigilant attention, kind of open-ended attention for the predators, they see that the animals use their left eye more to look for predators, which suggests right hemisphere dominance for vigilance. I mean, that's kind of the idea here, is that there's a lot of studies that indicate that the right hemisphere is particularly important for vigilance, looking out for potential threats. The left hemisphere, as the master and emissary suggested, is kind of important for focusing in on things, especially for the purpose of getting food. Right hemisphere, that open, vigilant attention. More evidence right there. And then this is just, I'm just gonna, the title of this article, it's a meta-analysis, right hemisphere dominance for vigilance. This kind of supports some of the claims made in Master and Emissary, in particular, that the right hemisphere is particularly important for vigilance. So it's another set of evidence that suggests the right hemisphere is important for vigilance. That open attention, waiting for something to happen, that involves the right hemisphere more than the left. Yeah, and then two more little things here, then we'll call it a day, actually. And then we'll start chapter 8 next time. So one more type of attention. We've talked now about spatial attention. That's the main one we've talked about. We've also talked about feature attention, when you pay attention to either the color of something or whether something has motion or not. That was type of feature attention. And then here is object attention, is illustrated in this study. So in this study, this little thing right here, this is an object right here. And this is another object right here. The idea of object attention is that when you pay attention to a object, you kind of have your attention spread over the entire object. Like you pay attention to a thing, and part of the way that what happens when you're paying attention to a thing is that your kind of attention kind of envelops the spatial arrangement or the places in space that object exist. And that this causes you to facilitates or improves processing of all the points in space that are touching an object. So it's probably easiest to just kind of show, I don't actually care about the brain findings for this one. I just care about the behavioral findings, which are right here, reaction time. And so here, this is a cue right here. And then the target would be something that occurs inside right here. And so this is a valid trial because the cue points to right here. And then a target occurs right there as well. So the cue is telling you exactly where a target occurs. You respond to a target when it occurs right there. That's this red condition right here. The reaction times are small. They're quick right there in the valid trial. But here we have another type of trial where here the target still occurs over here, but the visual cue is kind of misleading. The visual cue is telling you, pay attention right here, but then the target is occurring right here. So this is in a sense invalid because you're told to point to one side of space, but the target actually occurs on a different side of space. But in another sense, it kind of is valid as well, because here the target, or the cue rather, is pointing in this direction, but the target occurs as part of the same object. The cue points right here, and this point in space is connected to the other point of space over here by being connected to the same object. And you do see in this situation that there's a little bit slower response time, which makes some sense because the participant is not paying attention to the exact side of space that the target occurs. The target occurs right here. The participant is paying attention over here. So they're going to be a little bit slower. But what's really crucial is to see the difference between the green and the blue lines right here. And so this last condition, the blue trace right here, the blue line, blue bar, the cue is telling you to pay attention up here. The target occurs up here, but please notice now that these 2 points in space, they're no longer connected by being a part of the same object. It's physically the exact same parts of space that you are just paying attention to. You're paying attention to right here, the target occurs in this, you're paying attention to the upper right-hand quadrant. The target occurs in the upper left-hand quadrant. Here, in this one, those 2 quadrants, upper right, upper left, are connected by being a part of the same object. Here, though, in this third condition, you're paying attention to the upper right quadrant again. The target occurs in the upper left, but now these 2 points in space are not connected by being a part of the same object because the objects are now up and down instead of left and right. And that's this blue trace right here. And so what you can see is that even though you're attending to the same side of space here, and the target occurs in the same side of space here as that you do right here, paying attention to this side of space, target occurs over here, you're actually a little bit faster to respond to the target here in this condition, the green one, than you are right here, this condition, the blue one. And the reason is because in this condition, you benefit from object attention. These 2 points in space are both connected to the same object. When you're paying attention to this side in space, your attention kind of naturally spreads around all of the object, which facilitates your processing of things way over here a little bit by the fact that they're connected. It's connected to the object. Over here, though, you don't benefit from that because these 2 points in space are not connected by the same object. So you benefit from object attention here, but you do not benefit from object attention here. And that causes the green reaction time to be a little bit faster than the blue reaction time. And then this is the last thing, which is kind of fun, I think. Some classic feature integration theory. Kind of the main terms here. This is another behavioral paradigm. We're not going to really talk too much about the brain for this one. This is classic behavioral paradigm for studying attention. The main thing about this feature integration theory is the difference between a pop-out search and a conjunction search, which is illustrated right here. So a pop-out search is when you're looking for some target. So all these tasks, you're searching for some target. And in the pop-out search task, the target, which here is the red O, it has a property which is completely unique to it. So obviously right here, the property that the O has that's completely unique to it is its color. Its color red is unique to it. And so when it pops out, it's very easy to find. And if you look at the amount of time it takes to find the target in such a pop-out search, the reaction time tends to be small. And it doesn't matter how big the distractors are, how many distractors, that is like the amount of things around it. If the target you're looking for has a feature which is unique to it, you'll be very quick to find the target. It should pop out. Contrasting that with a conjunction search, which is right here, the same target, a red O, but in a conjunction search, there are other distractors around it, and those distractors around the target, and those distractors share at least one property with the target. The target, in other words, here does not have a property which is completely unique to it. If you look at the other green O's, they share the property of shape with the target. This O it's an O, and these other O's, they're also O's, so they share the same shape, O-ness. So the target does not have shape as unique to it. But the target here does not have its color unique to it as well, either because it's red, but then there are other red things as well. So these distractors out around the target, they share either the color or the shape with the target. And that's a conjunction search. When the target does not have a feature which is completely unique to it, then reaction times to find the target is a little bit harder. So let's do a couple practice with these and then we'll call it a day. So here would be a task, and the task is find the target, which is K. So would this be a pop out or conjunction search? Go ahead and type it in. Is this a pop out or a conjunction search? Let's take a look. Yep, it is a pop out, and the target here, K, has a unique shape. K versus R is the only thing shaped like a K. So that's an easy one. It's pop out. Let's do, yeah. Here is another example of a pop out search because we have a T here, which it has the unique feature of color. It is the only gray T in a backdrop of purple Ts. And so this would be another example of pop-out search. It's easy to find the target because it is the only one with that color. Let's see. Yeah, let's do this one. So the target here is find the horizontal 8. So is this one a pop-out search or a conjunction search? What is this one? Pop-out or conjunction search? It is conjunction, and I see a lot of people are getting that right. Can you explain how you knew that? How did you know that this is conjunction search? It's a little hard. You're right, but I'm curious to know how you guys can justify that, or how you can explain that you're right. How do you know it? You're 100% right. It is a conjunction search. In fact, the target is the horizontal 8. This is the target right here. It is a conjunction search, and it's a little bit harder to find this target here. But why is it? Why is that the correct answer? Why is this a conjunction search compared to a pop-out search? Okay, so let's see. The target doesn't have different feature because the 8 shares features with the distractors, because there are other distracting 8s and horizontal S's. That's a good one, that last answer in particular, because there are other distracting 8s. So first, let's look at this. The shape of this thing is 8. The shape is 8. And you can see that there are other 8s here. So this is the target. It does not have its property of 8ness as unique. It does not have 8ness unique. The other property that it has is horizontalness, and it does not have horizontalness unique as well because there's a whole bunch of other horizontal objects around it. And so this is correct, a conjunction search, because it does not have either property 8ness or horizontalness unique. It shares that property with other distractors. Therefore, the target doesn't just pop out. This is a conjunction search. We already did the in-class discussion questions, so that is it. We will start chapter 8 next time, and I will post that quiz for the last quiz. I think chapter 11, I will post that this afternoon.